So basically what has happened for the cell to get to early prophase, as you can see in the picture, is that interphase has taken place. Interphase accounts for about 90% of the cell cycle. During this phase, there are 3 stages, which we don't really go into detail about, but except for the second gap phase, which during this phase, the chromosomes condense into chromatids in preparation for division, and the centrosomes are replicated. And then after this, it goes into early prophase.
We are now in early prophase. There are 3 stages. So in early prophase, the nuclear membrane disintegrates, the nucleolus disappears, and the chromatin condenses into visible chromosomes.
This picture is showing late prophase, and during late prophase, the chromosomes continue to coil and appear as double chromatids. The chromatids are each joined by a centromere in the middle, and the centrosomes move to opposite poles of the cell. As they do so, they form the spindle fibers between the poles. During metaphase, the chromosomes become aligned in the equator of the cell.
We are now in early anaphase. So what happens is that the chromosomes are pulled to the opposite poles of the cell. The centromeres divide, freeing the 2 sister chromatids from each other. Each chromatid is now considered to be a chromosome. The spindle fibers thus begin moving to the once joined sisters to the opposite poles. We are now in late anaphase. The chromosomes have moved to the opposite poles, and what has happened is that each centrosome has pulled the chromosome towards it. By the end of anaphase, the 2 poles of the cells have equivalent and complete collection of chromosomes.
Telophase is basically the opposite of early prophase. During telophase, there's a formation of 2 nuclei. The daughter nuclei begin to form at the 2 poles of the cell where the chromosomes have gathered. The nuclei reappear and the chromatin becomes less tightly coiled